Hello everyone, I am Dr. Sakshi Sethia and this is my YouTube channel. I upload videos pertaining to pediatrics and pediatric cardiology. Please subscribe to my channel for regular updates if you haven't already. Today we'll be discussing about pyridoxine or vitamin B6. Why I have put this picture over here? It's because pyridoxine is mainly concerned with amino acid metabolism, and those with a very high protein intake, they tend to develop a deficiency state and should therefore take adequate RDAs for the same. Banana being a very rich source of vitamin B6. We'll discuss about vitamin B6 systematically, however, under the following headings. A tangential biochemical overview, who all develop a deficiency and how they manifest, labs, dietary sources, and finally the treatment of a deficiency state. Pyridoxine, it is used in the form of PLP, also known as pyridoxal 5-phosphate, which acts as a cofactor for many reactions. It is needed for amino acid metabolism, as I've already mentioned, in the synthesis of various neurotransmitters like GABA, glutamate, dopamine, etc., in glycogenolysis, and it modulates steroid action. We'll learn about these along with their clinical significances in the coming slides. A very important reaction in amino acid metabolism is known as transamination reaction. In a transamination reaction, an amino acid loses its amino group to a keto acid. In turn, this amino acid itself becomes keto acid. This amino group, when it is gained by this keto acid, it converts into another amino acid. So there is amino acid 1 which loses amino group and becomes keto acid 1, and keto acid 2 which gains the amino group and becomes amino acid 2. This particular reaction requires an enzyme called transaminase, which in turn requires PLP as its cofactor. For instance, in this particular reaction we have alanine. This alanine basically loses its amino group and becomes pyruvate, which is a keto acid. And this amino group is in turn gained by glyoxylate, which is a keto acid, and it becomes glycine. Now this reaction, a standard transamination reaction, it requires PLP as its coenzyme. If at all there is a deficiency of PLP or a deficiency of pyridoxine, this reaction will not happen. All the accumulated glyoxylate will not be able to convert into glycine, and it will in turn take another pathway and it will convert into oxalate, and there will be an excess of oxalate resulting in hyperoxaluria. And this hyperoxaluria basically presents as renal stones or bladder stones, and that is therefore one of the manifestations of deficiency of vitamin B6. Another important reaction in amino acid metabolism is the synthesis of cysteine. Now, cysteine is formed using serine and homocysteine. If you compare the structure of cysteine and serine, you will notice that serine has a hydroxyl group, whereas cysteine has a thiol group. This thiol group is derived from homocysteine, which in turn gains it from methionine in a reversible reaction. So, the thiol group is basically achieved from homocysteine, which gains it from methionine, and the carbon skeleton and amino group is derived from serine. And therefore, cysteine is synthesized using serine and homocysteine, which combine to form cystathione, and cystathione in turn degrades to form cysteine. Now, these two reactions basically require PLP as their coenzyme, and if at all there is a deficiency of PLP or pyridoxine, there will be something known as homocysteinuria. Now, what is to be noted is that B6 deficiencies is one of the causes why homocysteinuria develops, because there are other causes as well. If you look at the second panel, in these reactions, you'll notice that vitamin B9 and B12 deficiency also cause homocysteinuria. If you look at this particular reaction, if at all there is a deficiency of methylcobalamin, homocysteine will not be able to convert into methionine, and therefore will develop homocysteinuria. And also, if at all there is a deficiency of methylcobalamin, 5-methyl tetrahydrofolate, which is the inactive form of folic acid or vitamin B9, it will get trapped in this particular form and it will not be able to convert into tetrahydrofolate, which is the active form. So, B12 deficiency will lead to folate being trapped in an inactive form. Again, if there is a primary deficiency of folic acid or vitamin B9, again this reaction will not occur, and therefore vitamin B12 deficiency, vitamin B9 deficiency and vitamin B6 deficiency, all of these will lead to homocysteinuria. Be it any of these deficiencies, an excess of homocysteine will in turn convert into S-adenosyl homocysteine. And again, this is also a reversible reaction. If at all there is an excess of S-adenosine homocysteine, this reversible reaction will not proceed in this direction and will not form methylated products. Now, these methylated products are very important. Why? Because we have myelin basic protein which is being formed. If myelin basic protein is not formed, then we will not have myelination, or we'll have a demyelination disorder, which will manifest as peripheral neuropathy. To sum up, vitamin B6, B9 and B12 deficiency will lead to homocysteinuria and also lead to demyelination and peripheral neuropathy. If you remember from the previous lecture on niacin, I had mentioned that niacin is either ingested as niacin or it is synthesized in situ using ingested tryptophan, wherein 60 milligrams of tryptophan they are equivalent to 1 niacin equivalent, or 1 milligram of niacin. And therefore tryptophan metabolism is basically something which forms niacin, and it requires PLP. And a deficiency of PLP will lead to deficiency of niacin, leading to features of Pellagra. Also note that xanthurenic acid, it is a byproduct in niacin synthesis, and a urinary xanthurenic acid assay, it aids in diagnosing pyridoxine deficiency. Next, we have heme synthesis. The very first step in the formation of heme is delta ALA synthesis, and this reaction of delta ALA synthesis, it requires pyridoxine in the form of PLP. If at all there is a deficiency, it will manifest as microcytic hypochromic anemia. Then we have glycogenolysis. In this, glycogen phosphorylase is the one which requires PLP. Lastly, PLP, it binds to steroid hormone receptors, and it inhibits or prevents the steroids from binding to the steroid hormone receptor and prevents the transcription. Therefore, pyridoxine basically downregulates the steroid-mediated gene expression. To summarize, pyridoxine deficiency, it affects glycine metabolism leading to oxaluria, which presents as bladder and renal stones. Homocysteine metabolism is affected, leading to homocysteinuria, manifesting with the features of homocysteinuria, which are vascular features or soft tissue features, and also peripheral neuropathy. Tryptophan metabolism is affected, leading to Pellagra-like features, and heme synthesis is affected, leading to microcytic hypochromic anemia. Who all develop a deficiency state? Very predictably, those with a high protein intake, those on certain medications that inactivate pyridoxine, for example, isoniazid, penicillamine, phenytoin, carbamazepine, and others like oral contraceptive pills and even the corticosteroids. Then those on maintenance dialysis, they can develop a deficiency state. And lastly, metabolic deficiency or dependency states will have a manifestation. These we'll discuss in detail in the coming slides. A very important metabolic dependency state is pyridoxine dependent seizures. These seizures, they can occur in two scenarios. The first is that the mother had been given very high doses of pyridoxine antenatally, and the child now becomes dependent on its metabolism for very, very high doses of pyridoxine. Upon being born, when the high doses are not being provided, the child starts showing seizures, and these seizures can be managed with an interim high dose pyridoxine for a couple of weeks until the child's threshold normalizes. The second case scenario is an autosomally recessive condition which is inherited inborn error of metabolism. In this, the child can start throwing seizures as early as intrauterine life. But most usually, these seizures begin a few hours after birth, but sometimes they can also happen after the neonatal period ends, in the post-neonatal period. Usually, these seizures are multifocal, clonic type of seizures. But sometimes, even GTCS, which is a very, very rare entity in neonatal convulsions, but this can be found in pyridoxine-dependent seizures. So, GTCS can also be seen. These seizures are associated with neonatal encephalopathy, which can be characterized by hyperalertness, irritability, tremors, emesis, which basically happen before the onset of seizures. These kids, they usually have recalcitrant seizures, which however can be maintained on monotherapy of pyridoxine. Sometimes it can happen that the child was initially responding on multiple anticonvulsants and later stops responding to them. So we should give a trial of pyridoxine in such patients. Whenever suspected, a therapeutic trial of mega doses of pyridoxine should be given. If at all an IV 50 to 100 mg of pyridoxine is able to abort a seizure, and this is one of the diagnostic aid that the seizure is pyridoxine dependent. But please note that cessation of seizure usually happens immediately. But EEG changes, they may take a while. They happen maybe after, they basically resolve after a few minutes, or sometimes they may take even a few hours after giving the dose. But seizure termination usually happens immediately, and therefore it is a diagnostic aid. If you look at this particular EEG, you'll notice that there is asymmetry. And there is also asynchrony in the background, which is suggestive of a disorganized background EEG. This disorganized background EEG, it later ends up throwing spikes and slow waves. So there is a generalized, in all the montages, we are seeing spikes, slow waves, sometimes sharps are also seen, polyspike pattern is seen. So this generalized abnormality, it was basically because of pyridoxine deficiency, dependency, and this EEG had normalized after a few minutes of giving pyridoxine IV 100 mg. And here we can see a normal sleep pattern of EEG. Now, why does it occur? It occurs because of defect in metabolism of lysine. This is a very important point, and it has been asked many times in SS entrance that lysine metabolism is affected in case of pyridoxine dependent seizures. What happens is that lysine metabolizes to form a product called alpha aminoadipic semialdehyde. Let's call it alpha ASA. This alpha ASA is supposed to further metabolize or catabolize using this enzyme alpha ASA dehydrogenase. In case of pyridoxine dependent neonatal convulsions, this enzyme is deficient and therefore alpha ASA accumulates. This accumulated alpha ASA, basically it combines with PLP. It forms certain other compounds, and these other compounds, they combine with PLP and consume it, causing a deficiency or shortage of PLP. So this PLP is getting consumed because of these other products which have been formed in this particular condensation reaction. PLP gets consumed and we have a deficiency of PLP which happens. This PLP was required or needed in the synthesis of GABA, in absence of which we do not have GABA, right? So in absence of this PLP, which was primarily deficient because of absence of this enzyme, right? So in case of, when GABA deficiency develops, the child starts throwing seizures. That's all about pyridoxine dependency. Now coming to clinical features of B6 deficiency in older kids. They usually manifest with glossitis, cheilitis, and seborrheic dermatitis around eyes, nose, and mouth. This happens because impaired amino acid metabolism, it leads to dense or turbid ground matrix. Because amino acids are not getting metabolized, the ground matrix becomes turbid, which acts as a pro-inflammatory substrate, leading to inflammation. Now, peripheral neuropathy, kids don't usually manifest with peripheral neuropathy, adults however do, and it happens because of impaired methylation, as I've already explained, which leads to impaired myelin formation or demyelination, which, as I had already explained, happened because of accumulation of S-adenosyl homocysteine. Other manifestations are oxaluria leading to kidney stones, bladder stones, homocysteinuria, which manifests with vascular features or other soft tissue features, then pellagra and microcytic hypochromic anemia. For diagnosis, since PLP is required in transamination reaction, the activity of transaminase enzymes being low will be indicative of a deficiency state. So we have decreased activity of hepatic transaminases, AST and ALT, it indicates a deficiency of pyridoxine. And a urinary xanthurenic acid assay will also be aiding a diagnosis of pyridoxine deficiency pertaining to altered tryptophan metabolism. For non-vegetarians, meat, fish, poultry, and liver are rich sources of vitamin B6, whereas for vegetarians, it is rice, bananas, and certain other fruits and vegetables. Coming to treatment, for seizure termination, high doses in the order of 50 to 100 mg of pyridoxine, which should be given IV, and sometimes even more than 100 mg is required to terminate a seizure, which should be followed by a monotherapy in case of a dependence state. This monotherapy should be given every day, lifelong. If it is given IM, the dose is 2 to 10 mg. Per oral, the dose is 10 to 100 mg, on how much ever dose the child is being maintained. Then toxicity. Pyridoxine is one such vitamin which causes peripheral neuropathy in deficiency as well as in excess. So, even toxicity is leading to peripheral neuropathy. This usually happens in those who are not metabolically dependent and are still iatrogenically being given high doses in the order of 100 mg. So that's all about pyridoxine. Thank you.